Hi everyone, I hope the session is being useful for you. I'm Janet, and I'm a postdoctoral researcher at the research program on Biomedical Informatics, which is affiliated to the Hospital del Mar Research Institute and Pompeu Fabra University. I hope that some of you here today at least have heard about DisGeNET, and maybe some of you already have used it in your research, for example. And my expectation would be that after these 45 minutes, you will learn something new about DisGeNET and how to use it better. And those of you who don't know, who haven't heard of it, then I can convince you to use it in your teaching materials or in your own research. As you know, the genomic it's making a transition from the research lab to the clinic environment because of the new technologies, and then because our increased capacity of finding and assessing genetic variation, and also the many tools that have been developed to evaluate the impact of this genetic variation, for example, in health, right? And you can use this information, genomics information, not only as Sergi was discussing for diagnosing patients with diseases, but this genomic information can be also used for tailoring treatment, to evaluate or predict the patient's response to treatment, but also to stratify patients, or for biomarker discovery. Also, there is a new field of polygenic risk scores in the field of GWAS data. And yes, finally, in drug development, genomic is playing a role every day that is more important. This example of this is a recent publication that was stating that 2/3 of the FDA approval from 2021 had some type of genetic information related to the target of the drug, and we even state that this percentage is actually higher. So a lot of drug development today is being guided by genetic mechanisms. So this is very good, right? But then to use genomic data, and this is also true for other biomedical data, researchers have to face different challenges. Like, for example, the fact that the data is scattered across different silos, the lack of interoperability, the fact that the innovation is locked in publications, and the large volume of information. Given that I have more time today to explain, then I would be explaining each of these challenges separately.
Can I just say, sorry, Janet, I don't know if you're sharing slides because we're not seeing.
Yes.
I wasn't sure. Yeah.
Okay. So I will just, thank you, Amy.
No, it was a question in the chat. I didn't know if you were just giving an overview first. Okay, well, now we're seeing.
Up to so far, it was an overview, but now, yes, like now, can you see my screen?
Yeah, now we're seeing presenter mode. Perfect.
Thank you. So now I was talking about the different challenges, and I was saying that the data is scattered across different silos, but Babita, I think, was also referring this in her earlier presentation. So everyone that wants to understand different the mechanisms of diseases, let's say, for example, the genetic basis of human diseases, you will have to search in different repositories to find all the information. This happens for Mendelian diseases or for complex diseases. Also, Babita was also mentioning earlier about this Babel Tower, in which everybody calls the same thing in different ways, right? So this is an example that I searched that is a little bit extreme, but it's to illustrate my point. So here, for example, for the case of the gene, the same gene, which is lipocalin-2, can be mentioned in publication in these many different ways, right? Sometimes when people come from the genetics field, they name genes in one way, but if you come from toxicology, then sometimes you refer to the same gene in another way, which makes it very difficult to integrate all this information. And we have a similar scenario for variants and also for diseases. For example, Wilson disease is also called hepatolenticular degeneration, and these are varieties of the same term. So this makes it difficult to integrate the information. This is for terms, right? But the same panorama or similar panorama we find if we are using vocabularies. Like in the case of genes, we have different ways, so we can use the official symbol, right? But we could use the interest gene identifier or the HGNC, encelmet genes, uniprot accessions, etc. And for diseases, this I think is even worse because we have different types of disease vocabularies. Sometimes they are super specific, like for example, the disease ontology, which be more focused on monogenic diseases, and the ordo, which is of use by Orphanet, which is a catalog of rare diseases, right? So, and sometimes, yes, some vocabularies would not cover all diseases. So this is the second challenge that we have to face when dealing with human disease information. The third challenge was that the innovation is locked in publication. That means that even when we have many databases that store information about the genetics of human diseases, or which is variants are associated to disease, most of these databases are the data there come from manual curation, from, for example, from scientific publication, right? But this is a task which takes time, and so a lot of information that is recent is not covered in these databases. So if you want a full panorama of every possible gene associated to your disease of interest, then you have to go to the scientific literature. But we also know that there is a lot of scientific literature published every day. It's estimating that you have around 8,000 papers a month publishing in the field of disease genomics. And for some diseases like Alzheimer's that are very heavily investigated, but you have to keep up with a lot of new publications. So if you want to have, again, the full panorama, you will need to recur to text mining approaches to be able to digest all this new information. And finally, well, we are happy because we have a lot of patients that have been sequenced, because the cohorts of patients, for example, that have been studied using GWAS technologies are increasing every day more. So this is a very nice picture that summarizes our technological advances from the last decades, right? So the increase on, for example, genes that are associated to monogenic diseases, the number that have increased a lot in the last years. Even we have 4,000 of genes associated to rare diseases. We have right now in the GWAS catalog, if you search and then you filter by significant variants, we have 100,000 of significant variants associated to different traits. If you go to ClinVar, then you have over 1.5 million of variants that have been deposited there. So yes, we have a lot of information, and this is good, but also we have to find ways of dealing with this large volume. And this is why we decided to develop DisGeNET, which was like 10, 12 years ago now. That is a database that integrates information about the determinants of human diseases, the genetic determinants of human diseases. We also want to highlight that we cover the full spectrum of human diseases, but also we have information about phenotypes and traits, and about drug adverse events. And given that we annotated the data with different vocabularies, we are an interoperable resource that supports different applications in genomic medicine and also in drug development. So a little bit more details on how we develop DisGeNET. Well, as I mentioned, we integrate the information from curated data sources that does a different nature. Like we have two different GWAS repositories, GWAS catalog and GWAS DB. We integrate information from animal models, from the mouse and the rat genome databases. We have information from rare diseases from Orphanet, from the panel lab, from Genomics Inland Panel Lab, from UniProt, from ClinVar. And we also, this information, we are integrate with the data that we extract from the scientific literature using natural language processing tools. And well, this at the end results in more than 1 million relationships between genes and diseases, and 300,000 variant disease associations comprising this many genes, like 22,000 genes. So we almost have information for disease association for almost every protein coding gene. And we have information from 30,000 diseases and traits. I want to highlight here that, again, that we have diseases for every, we have information about genetics of diseases belonging to different therapeutic areas, like, for example, most of them, well, as you can see here, mainly congenital hereditary abnormalities, but also for nervous system diseases, obviously for cancer, and yes, metabolic diseases. So we are representing every therapeutic area. The information stored in DisGeNET currently is equivalent to 800,000 publications. But notice that the 70% of this, the relationship between genes and diseases are extracted using text mining approaches. So these are not present in the 15 or more databases that we integrate. So we need text mining approaches, as I was saying before, right? And given the importance that the text mining has for DisGeNET, I wanted to take the opportunity that we have a little bit more time today to explain a little bit more about the biomedical text mining that in general, but also the one that we do in DisGeNET. So the text mining, the biomedical text mining usually starts with the information retrieval steps in which you keep only the important documents. In our case, we start with all publications, scientific publications in Medline, but we perform a query that allows us to filter, to keep only publications that are related to disease genomics in human. We then perform a step of name entity recognition in which we detect mentions of genes, variants, and diseases in the abstracts. It's important. This is in the abstracts of the publications. And this name entity recognition step is usually based on dictionaries. So we have a dictionary of genes, of every possible gene, and how these different genes can be named, and the same for diseases and variants. And afterwards, we perform a relation extraction step that uses machine learning approaches, specifically super vector machine, that at the end is able to detect relationships between genes and diseases and various diseases in the abstracts of the scientific publications. This information is stored in DisGeNET, but also can be used for knowledge discovery, right? This seems simple, but it's not. So and why is this complicated? Because it's the ambiguity inherent, for example, to the names of the genes. So not only to the name of the gene. So this, for example, here, an ACT can refer to neoadjuvant chemotherapy, for example, but also can refer to two different genes, N-acetyltransferase and sodium coupled citrate transporter, right? So this is one problem that doing text mining has. A second problem is that the same thing can be referred to in many ways. Like for example, here you can do, for example, if you're trying to catch the relationship between this and this entity and this entity, there is many different ways in which a scientist can describe the relationship. We see difficult the extraction, for example, of the relationship between these two entities, right? And also, these are examples that I'm taking from DisGeNET. Sometimes doing text mining for genes and diseases is difficult because the scientists prefer, because sometimes the name of the gene coincides with the name of the disease that the gene causes, right? So this is the case for neurofibromatosis type 2, right? That the gene is also called the same. So here, the only way that you can deal with this is going beyond the name entity recognition step, right? And training a machine learning classifier to detect the different contexts in which a disease is mentioned and a gene is mentioned, right? This is another example for the Duchenne muscular dystrophy. And the third example here is an example that we have not been able to solve yet, that is related to the sentences in which different pairs of gene diseases are mentioned, right? So here you have, and they have identified significant interaction between these SNPs in these genes in the context of lung cancer, but also in the context of non-small cell lung cancer, these genes are important, right? So my point is that here we are currently not being able to separate. So we will probably have this information in the database, like these genes are associated to lung cancer, to non-small cell lung cancer, and squamous cell carcinoma, right? And the same for this. So in this case, we are mapping every possible gene to every possible disease, which is it's a handicap for the text mining methodology. So how do we use text mining in DisGeNET? We use it mainly for three different tasks. One of them is, as I mentioned before, identifying the mentions of genes, variants, and diseases, and the relationship between them, the classification of gene-disease associations, and also for detection of negations in the sentences. So this is an example of the first task. So you have these two nice sentences here. And after the name entity recognition step, we will be able to identify this term here, Alzheimer's disease, like a disease, and we will normalize this term and standardize it to this concept unique identifier, which is the vocabulary that we use in DisGeNET that comes from the UMLS, the Unified Metathesaurus Language System. And then we are also able to detect the mention of this gene here, amyloid beta precursor, and then we are able to normalize it to the gene interest identifier from the NCBI, which is the main gene identifier that we use in DisGeNET. And we are able also to mention, to standardize the mention of these SNPs to this DSNP identifier. In the next steps, then we will run a machine learning classifier, and we will be able to detect the relationship between this gene and this disease and the variant and disease. So for the second task, which is the classification of gene disease association according to the DisGeNET association type ontology, we rely on a gene disease association type ontology that was developed also in our lab to classify different types of relationships between a gene and a disease, right? Because not every gene will be mentioned in the context of causality. So not every sentence that you will find in DisGeNET will be reflecting the fact that the gene is causing the disease, right? So depending on the type of disease, you are never going to be able to say this, because we know that, for example, in the context of complex diseases, where we have some genes that increase the susceptibility of the person to suffer the disease, right? And then we are like the causal mutation is more reflecting the case of Mendelian diseases like Duchenne or cystic fibrosis, in which having a mutation implies that the patient would develop the disease, right? So we try to reflect all these type of different relationships, or for example, a gene that is used as a target for a drug that treats the disease, right? But this gene doesn't have to be necessarily a gene causing the disease, but it might be a gene that is downstream the perturbed pathway of the original cause, right? So this is why we design, for example, the therapeutic relationship between gene and disease. So as I was mentioning before, we have trained our machine learning classifier that also not only identifies relationships between genes and diseases, but it's also assigns one of different of these possible association type. And I wanted to show so how does this look like a little bit. So for example, in the case of alter expression, you will see that the gene expression correlates with the phenotype, or you have mRNA ratio of the gene altered. So the typical phrase would be this gene, the expression of this gene is altered, it's perturbed, it's increased, it's decreased in the context of the disease. For the case of post-translational modification, we mainly have phosphorylation, so genes that are phosphorylated in the context of the disease. And finally, for example, here for susceptibility mutation, we have a variant that increases the presence of a variant in a gene increases the risk for a particular disease, right? And the third case is the detection of negative statement. I don't know if you have used DisGeNET before. Maybe you have never noticed that we have in DisGeNET something that we call the metric that is called the evidence index. That the evidence index characterizes a relationship between one gene and one disease, or one variant and one disease. And it quantifies the number of publications that have a positive assertion saying that this gene is related to this disease, divided by the total number of publications. So an evidence index lower than 1 would indicate that some publications are stating that the gene is not associated to the disease, or the presence of a variant has not been is not increasing the risk for the disease. You have several examples here in the slide. So there was no association between this SNP and ischemic stroke, for example, no significant genetic factor 4, etc., etc. So yes, this is another thing that we also have in DisGeNET. So as I was mentioned before, if you have a large volume of information, then it would be useful to find ways to rank and order the information, and also metrics to filter the information. So you can, like in the case of Sergi was explaining earlier, so you can actually, when you have many hits, pues go and reduce these hits to a number that is more manageable, for example. So this is why in DisGeNET we have different annotations. On one hand, on the other hand, we have different annotations for the entities, like for example, disease, genes, and variant to ease the interoperability. So you can always query our database with your own identifiers like HPO or IMIM identifier or mesh identifier, so the one, your favorite one, and you are able to retrieve the information. So this is the case for the disease annotations, in which you have different vocabularies, but also we provide the semantic type and the DisGeNET type. The DisGeNET type for diseases allows you to filter for example, filter out things that are not properly diseases, but are more like phenotypes and traits if you only want to keep only the diseases, or vice versa. So the DisGeNET types cover 3 different categories: is the disease, group, and phenotype. And then you can use it to filter the results of your query, right? And the same for the genes. So we provide different attributes for the genes, like the symbol, the name, the protein class, and for the variant, we have the consequence type. If we have time at the end, I can show you all these attributes via the web. And we also have some particular metrics that are specific for DisGeNET that allow you to, like, for example, filter the genes according to, and the variants according to the pleiotropy or the specificity. And for the gene disease associations, we also have some attributes and metrics, like I explained earlier, the evidence index. There is also the evidence level that we take from different curated databases like ClinGen and the Genomic England Panel App. There is DisGeNET association type that I just explained, and the number of articles, the number of publications and SNPs characterizing the gene or the variant disease association. Finally, for each gene disease association, we also keep the provenance of the original statement, let's say, so you can always trace back the information to the original source, either a publication or even the original database from where it was extracted, right? So how can we use DisGeNET, or what type of use cases you can have? For example, if you're a teacher, if you have a list of variants that are coming from, for example, for a GWAS study, you can use DisGeNET to contrast if these GWAS variants have been already detected in some other studies like GWAS studies, or even in the scientific literature. Sorry, or you have results of something like Postdoc was showing before, and then you want to see if these variants have already been detected in the context of the disease that you are querying, or even in the context of similar diseases. You can use different things that we have developed in DisGeNET that I will show, I think, in a few minutes, to find if the variant is also associated to semantically similar diseases than the one that you are studying. You can use DisGeNET, for example, to develop disease-specific gene panels, because you can use DisGeNET score to filter and say, well, I'm interested in developing a disease panel for autistic disorder. You can use the score to rank the genes and to keep, I don't know, top 100, top 200 of the genes that are in DisGeNET. DisGeNET can be used to identify new targets for therapeutic indications, as example that I was mentioning before at the beginning about the FDA drug approval. So many of the genes that have been that are the targets of the drug that were approved last year are also involved in the disease mechanism. In this case, you can see the example of utrophin, that is a gene that is not causing the disease, so it's not directly involved in causing the disease, but it has to do with the presentation of the disease, and it's a therapeutic target for Duchenne. Finally, you can use, for example, the information in DisGeNET to perform, for example, you have a target, so you have a drug and that has a series of targets. You can input this information in DisGeNET. For example, this is done with the DisGeNET package, and then you can compare and do a DisGeNET enrichment and retrieve a set of phenotypes associated to your list of targets that are, for example, might be related to a specific drug. Finally, the last a more recent and very exciting use case that we have been lately noticing that DisGeNET is being used a lot, and we are starting to explore this in the context of some projects that we are involved in. It's the integration of DisGeNET data in knowledge graphs. So typically a knowledge graph is built by integrating different types of data. Like, for example, well, these are 2 publications that use DisGeNET in the creation of a knowledge graph. One of them is very recent, that was a publication that was written by Patrick Alloy in the ERB, like I think last week or 10 days ago, and they integrate different type of databases, like, for example, pathway databases, gene disease databases, drug target database, drug indication, data sets, so that they integrate them. In any case, a knowledge graph would benefit from the integration of data sets that are often heterogeneous natures, biomedical data set, different types, and then you can use these graphs and using certain techniques to create embeddings. You can use them neural network approaches, for example, to train this graph to perform different tasks, like, for example, to suggest new gene disease association, or you can use them to suggest new indications for drugs. So if you want to access DisGeNET, you have different ways of doing this. First, you can go to the web interface. That is what I will be showing in a few minutes, because given that I didn't know if the audience what are the programming skills, I can show something that we can do via the web interface. But if you have you are a programmer, you can also access our REST API, in which you can reproduce every query that we can do via the web interface, and then you can integrate this our REST API endpoints to your own data pipelines, for example. We also have a SparkQL app API if you are interested in the semantic web. And if you like system biology, or you're teaching system biology, or you're teaching Cytoscape, I will strongly recommend then checking our DisGeNET Cytoscape app, which is available in the Cytoscape app store. But also you can check here, you have some examples of how you can use the Cytoscape app via R and create your own networks and do the same queries that you would do via the web interface. And finally, we have an R package. If you like R, you can use that package. And then we have a very extensive vignette here with every possible query that you can do, like retrieving genes associated to diseases, or the variants associated to the list of diseases, and doing multiple queries, etc., etc. Finally, I want to mention that, well, DisGeNET has been around like for over 10 years, as I mentioned earlier. So we have over 3,000 citations and well, 90,000 users. And yes, we are linked by different databases like UniProt, Reactome, and the Ensemble Variant Effect Predictor, etc. And well, given this success that we had, and given the fact that the data in DisGeNET cannot be used for commercial purposes, we also launched 2 years ago, into 2020, a company that is called My Bioinformatics Solutions, which has the purpose of developing DisGeNET Plus that have several advantages over the public version, the academic version, let's say, which is it has a higher frequency of all dates, and it has a better text mining performance, mainly because we are using deep learning technologies instead of support vector machine and traditional machine learning that we were using in DisGeNET. And yes, if you want more information, you can check here in the presentation and given the contact. And finally, before I go to the demo, to the web interface, I wanted to talk a little bit about the new data set that we have produced that has been also converted into a Shiny app that is called the Clinical Biomarkers app, that can be accessed from DisGeNET in this URL. And I will give a little bit more details. So the Clinical Biomarker app, as I see it, is a little bit of complementing the information that we have in DisGeNET, because the information that we have in DisGeNET is more like from the research point of view. So we extracted from scientific publications, but then the scientific publication might be dealing with patients, but it may be dealing with cell lines, or like animal models, etc. But we believe that if we do text mining of clinical trials, then we are then closer to the clinic, right? So this is why we did this exercise of doing machine and text mining approaches for clinical trials information. So here I am talking about the first step, which is data retrieval, right? So we use this aggregate content of clinical trials initiative, in which they have standardized and normalized a lot of information in the clinical trial, right? So they advance a lot in the way of being able to include the information of clinical trials in our automatic pipelines, right? So you cannot include free text. You need to process this, normalize it to be able to put this information in the context of the like a pipeline, or in the context of gene and variant interpretation, right? So they actually have done a lot of work in reflecting this in a normalized way. Like, for example, they have 54 tables in the database. This is this monster that you can see over here that has information, like normalized information for drugs, for conditions, for the study types. So there is a lot of information there, but there is no normalization of genes or proteins in clinical trials because let's face it, until a few years ago, the genes and proteins were not so frequently used in the clinical trial. This is more something that has occurred in the last decade, the last few years, let's say. So the first step was, after the data retrieval, was to examine all the sections that they have there to identify those sections that were more useful to our purpose. So we decided that the different sections of the clinical study, we were going to mine for genes, for the sections that were dealing with outcomes and with the design of the clinical trial. As I mentioned before, the condition information, it has already been normalized using text mining approaches by the people in the AACT. So we did not need to do text mining for the conditions, right? So this is an example, a couple of sentences that are examples of what we find in this type of sections in the outcome and outcome measurement. So if you are familiar with the type of sentence from a scientific publication, you will realize that there is a different way in which the statements are reflected here. So it's more like it's not the same way in which a scientific will write a paper. It's more like a telegraphic way, and this was a new challenge for our text mining system, right? So after we have identified the section of interest, we also perform an entity recognition. Here you have examples again of what we are dealing with in the outcome and the science section of the clinical trials. So here you have, again, different ways you have the same challenges, so you have different ways of mentioning the same entities. And but then notice that these are like nicer examples because they mention the disease, but many of them do not mention the disease. But again, we are not doing disease recognition in this step, only for the genes. Then in a third step, we train different classifiers using a random forest, so simple traditional machine learning approaches, to be able to assign these short statements that are reflected in the outcome and the design sections of the clinical trial, and say, well, the relationship between this gene and the disease in the context of this clinical trial belong to one of these categories. These categories are a little bit inspired in the original DisGeNET association type ontology, but they come from extensively checking all the types of relationships that were happening in the clinical trial. So we came up with these 6 categories. So I would now go to the web to show a little bit more about DisGeNET and the clinical biomarker app, unless you have any question, right? If you do, then you can ask me now, or you can ask me later.
We can feel free to ask a question. It's a quiet group.
Sí, I noticed.
Okay.
So I am here in the DisGeNET website, and yes, you can then, if you start exploring DisGeNET, of course, you can go here to search. And notice that you have 3 different tabs, right? Genes, disease, genes and variants, right? And then you can start by, of course, typing a disease name. Let's choose schizophrenia. And then you can click here, but if you also control click, you can actually select more than one schizophrenia subtype. Notice here that the fact that you use the UMLS concept unique identifiers allows us to have a lot of different subtypes and a lot of nuances for each disease, let's say, because this vocabulary is very rich and they integrate like a lot of different vocabulary. So you hit here, search, and then you have the results, right? So you have the 2 diseases that you search for here, and then you have several characteristics or several attributes for the disease that you queried, and then you have different buttons. So you can actually, if you say, well, maybe I want to see other similar diseases that I have, that I might have missed, you can go to similar diseases, This is a search that uses semantic approaches, so the relationship between the diseases. Here you have, for example, in yellow, these are terms that are common for the two diseases that you have queried, and in red is the second disease that you have queried. So let's say that you can also want to add paranoid schizophrenia, and then you hit spam search with selected diseases, and now you have the three diseases that you queried. So after that, you can, for example, go straight to explore the information in DisGeNET, and it doesn't matter what you click here, you would always be able to browse the information for each of these tabs, like they are here, if you can see them, yeah? You notice over here. And here you have the three diseases that you search for, and notice that you have source all. So here you have the preview of the results. So in the column, this column, you have the disease that you search for. Here, schizophrenia is the one that comes out first because it's probably the one that has more information. And the logic of the color here has to do with the entities, right? So in light blue, you will see attributes for the gene, and in green, you will see attributes for the relationship. So here you have the genes associated to schizophrenia with the UniProt, the full name, the protein class, and the numbers of all the diseases in which this gene has been detected, the specificity index, which is basically 1 divided by the total number of diseases and then normalized. So basically, if it's closer to 1 means the gene is very specific, is closer to 0 means that the gene is really pleiotropic and is associated to many other different diseases, which might be useful if you are, like, for example, trying to find a new drug target, right? You would want a gene that is really specific, but it's not associated to many other diseases. The pleiotropy index has to do with the therapeutic areas for the diseases to which the gene is associated to, right? So if a gene is associated to diseases that are falling all between, for example, the classes of mental disorders, so like typically dopamine receptors, they usually, even if they are associated to many diseases, if they are associated to the same disease class or therapeutic area, this would be higher, would be lower, I'm sorry, so the gene will be not very pleiotropic, but if the gene is associated to many diseases in different therapeutic areas, then the gene will be highly pleiotropic. And this is the probability of being loss of function intolerant taken from the NOMAD database. And regarding the association, we have here, for example, the evidence level that I was mentioning before, the evidence index that, as we saw in the presentation, it has to do with the how many or what is the proportion of publications that are stating that there is a positive association, or the way I see this, it's indicated that some publications are saying that there is no association, but as you can see, most of the publications here are accepting a positive association between the gene and the disease. Here you have the number of publications coming either from the curated databases or from the text mining approaches that are supporting the relationship between this disease and this gene, the number of variants that are associated to this disease gene, for example, between serotonin receptor and schizophrenia, right? So you can see here that there are 7 variants. And also something that I find really useful are the first ref and last ref that indicates that the first papers out of these 50 papers, the first one was published in 2001 and the last one was published in 2019. Meaning that this is an association that has been known for a long time, but the people still keep investigating it. So that means that it's an active, let's say, actively investigated association. And on the contrary, you have some others that have been investigated once in one publication or twice in 10 years, 20 years ago, and maybe this is a criteria that you can use to filter out information that you might not be really interested in, as if it's like this case that you have so many gene disease associations, right? So we have 2,900 results, so almost 3,000 results for only 3 diseases. So we have to find ways to prioritize and rank this information, right? So by clicking the links here, you can, for example, investigate what are the publications, exactly the evidences relating this gene, REL-N, and the schizophrenia, right? So you have here the result of clicking in the number of publications. So you can see here we have 87 results. Here you have the number of which is the original database that is producing these results. In this case, B3 is our text mining system. So this would indicate that we are seeing evidences that have been obtained via text mining. And here you have an example of what I was mentioning before, a negative association. There was no statistically significant association between the variant and the gene in the population, right? So you can, if you can still play a little bit more around here. For example, some of these are very recent. And then if you close this filter, then you will be able to explore all the associations for every gene in this tab that you saw in this tab, right? So if you remember, the first gene was AKT1. So using this, I don't know if you have noticed here that you have this hidden panel that can be also used to further filter the information. Like, for example, you want to say, oh, I have too much information. I want to see only the curated sources, right? So you can click there, and then you will see that the B3 is no longer there, but then you can see different relationships, different databases like CTV and DisGeNET, etc., right? And then you can say, well, I know that as an expert in the topic, I know that the schizophrenia is really related to GPCRs, and then I want to see how can I filter this and keep only the GPCRs. Well, you can use the protein class to filter the information and keep only genes that are GPCRs, right? But I still find that this is a lot of, you have now only 100 results to check, so you can check the results. But I find that always the summary view allows you to have a very quick bird's eye view of the results, right? So here you have the different GPCRs and the number of publications and the first ref and last ref. So I want you to notice now that when I close here to return to the old results, you will see that these numbers will have changed. And then you see that now we have all the papers coming also from text mining, right? And yes, now we will go to explore the, okay, I wanted to, so I know also, because I have been investigating a little bit about schizophrenia, that the dopamine receptor number 2, it's related to the treatment of schizophrenia. So maybe we can check here the information between that is supporting the variants related to the dopamine receptor 2 and schizophrenia. So here you have then moved to the summary of DDAs tab, right? And then you can see then the information related to the variant. So now you have in purple the attributes for the variant. So you have the gene in which the variant is located, that we already filtered out the rest, so we only wanted to see the ones that are associated to the dopamine receptor 2. But also you have a series of features that are annotation for the variants, as I was mentioning before, for example, the consequence type that is extracted from the variant effect predictor, and also as the same that Sergi was showing before in their resource, the allelic frequency in NOMAD and in the exomes and genome. So you can see that some of these variants are very frequent, except for these ones, these ones, and here are variants that are frequent in the publication, and then you also have the first ref and last ref. So maybe we can click, for example, here in this variant to see the publications that are related to the variant. And so, for example, if you explore the evidence here, you will find that this variant is related to responses to antipsychotic treatment in different samples of patients. So this variant makes that the patient responds better to treatment. What I did here for this use case was to combine the information from DisGeNET with the information that offers PharmaJKV, which is a database that contains information about pharmacogenomic variants. So sometimes combining different sources of information can be useful to better explore the data. And finally, you can also, if you want to find other diseases that are associated to schizophrenia, then you can go to the tab of summary of disease association, right? So what we did to produce this tab to find diseases that are related to your original disease query but it's not in the same sense in which we did at the beginning with the semantic search. So in this case, what we did was to compute the number of shared genes and shared variants between the disease of interest, in this case, schizophrenia, with other diseases, right? And then we computed a Jagged index, which is basically the union, the intersection of this between the union, divided by the union for the genes and for the variants. And yeah, we have ordered this by the Jagged index. And then here you have all diseases that share genes and variants with the schizophrenia, right? So here, for example, you have many diseases that are mental disorders or nervous system diseases, but you also have different types of cancer. Let's say that you are interested in only keeping those diseases that are mental disorders, because you are interested in checking other disorders that might be comorbid with your disease of interest. So you can use, for this filtering, you can use the disease class filter. And I would really, really recommend here that you use always contains, because this is mesh, this is taken from mesh disease classes. Mesh disease classes usually are associated more than one to one disease. So when you use the contain search, then you are saying, I want to keep every disease that is specified as a mental disorder, but I don't care that it's also classified as a nervous system disease, for example. Then you filter. As you notice here, you have many other filters that you can apply. In this case, I will only apply this one. And then you see here that the list, it's a little bit reduced, but it still looks a lot like the original one, because it's actually impressive the way that there's like they have a very high shared genetic component in this type of diseases. So, but when then maybe you want to say, well, I want to know schizophrenia and bipolar disorders and not to be highly comorbid diseases. There are some papers that even say that the schizophrenia and bipolar disorders are part of a continuum, where bipolar disorder is like a milder version, right? So you are interested in knowing with the shared variants between these two diseases. You can see here they shared over 200 diseases. And you can then explore the information about these variants, right? If you go here, you return to the disease information. And I think that my time is like a little bit more than we should. So I just want to remind you that, that you can have access to the API here, only that you need to register for having the API access and using the API, but it's only free, so you have to register for free, and then you can insert your key here and then use the API using your favorite programming language. And I think that I will leave the clinical biomarkers up for another time. But yes, thank you for staying so late and bearing with me. And if you have any question, I will be happy to answer.